Welcome to the Deep Dive, your shortcut to being truly well-informed. Today, we're plunging into, well, what feels like the most ambitious frontier of medicine and surgery, tissue engineering and regenerative therapies.
It really is. And, this field is moving incredibly fast. Things that seemed like pure science fiction just a few years back are now genuinely in clinical trials.
Right, so our goal today is to really extract the core science you need to get your head around this complex topic. It feels essential.
Absolutely essential.
Yeah.
Because it represents a pretty fundamental shift, I think, in medical philosophy. For literally centuries, the focus, especially for surgeons, was on tissue repair.
Repair. Okay, like what? Fusing a broken spine, maybe? Patching up a wound?
Exactly that. Or, putting in a graft. But now we're genuinely moving beyond just fixing or patching. We're aiming for true functional tissue regeneration.
I like that distinction. Repair often means, well, a compromise, doesn't it? You get a scar, maybe a stiff joint, something that isn't quite what it was.
Precisely. Whereas regeneration, as our source material defines it, is about these localized delivery strategies that actually mediate a full biological restoration, getting it back to how it was ideally.
Okay. And the core challenge driving this whole field?
It's that many crucial tissues in our body just have very limited, sometimes zero, capacity for spontaneous healing. Think about articular cartilage in your joints, or cardiac muscle after a heart attack, or large sections of your intestine.
Yeah, damage there often feels permanent.
It often is, leading to permanent functional problems. That limitation, that biological gap, is exactly what tissue engineering is trying to bridge. So our mission today is to break down this whole multidisciplinary area by looking at the three key pillars needed for regeneration: cells, materials, and molecules.
Cells, materials, and molecules. Okay, sounds like a solid framework. Let's unpack this. Where do we start? Maybe why is this so clinically relevant right now?
Right. Well, the clinical relevance is, frankly, staggering. Because regenerative therapies have the potential to completely change how we manage patients across almost every single surgical specialty.
So it's not just one niche area.
Not at all. If you just look at the conditions surgeons see every day, there's almost certainly an application, or at least a potential one, emerging from this field.
And the sheer breadth is what really strikes me. We're not just talking about, lab experiments anymore. We're looking at things that could help huge numbers of patients with chronic, often debilitating conditions.
Absolutely. Take the musculoskeletal system. Regenerative approaches are already targeting degenerative joint disease, traumatic injuries to bone, and crucially, articular cartilage.
Cartilage is a big one, isn't it? Once it's gone, it's pretty much gone.
It rarely recovers its original function. That's the problem.
Mm-hmm.
Then, moving to the cardiovascular system, you've got applications for massive problems like heart failure trying to regenerate cardiac muscle, or tackling vascular disease like atherosclerosis or aneurysms in large blood vessels.
Wow. And what about areas that seem even more complex to fix, like the gut, the visceral and GI tracts?
Major targets there, too. Think about insulin-dependent diabetes. If we could successfully regenerate or protect the functional pancreatic islets that make insulin, well, that changes everything.
That's a game changer.
Totally. Or consider patients with severe intestinal failure, maybe after massive surgery for Crohn's disease or trauma. Engineering a functional replacement for the small intestine. That's a huge goal, but it's on the horizon.
And you mentioned urology.
Yes, bladder reconstruction. Particularly for congenital problems, the ability to actually culture and implant engineered bladder wall structures using the patient's own cells is becoming a reality.
It's fascinating, even the respiratory system, you're saying.
Yep, tackling things like reconstructing the trachea or bronchus after cancer resection, or if there's stenosis, a narrowing. It's really clear the ambition is to step in wherever the body's own repair mechanism either fails completely or just gives you a subpar result.
So, tackling specific tissues now, but what's the really big picture, the ultimate goal?
Well, the ambition scales right up. While fixing cartilage or blood vessels is crucial now, the grand, long-term technical challenge is the replacement of entire, complex, solid organs. Things like the liver, the kidney.
Wow, that still feels a long way off.
It is. But the work being done now, perfecting how we culture cells, how we build scaffolds for simpler tissues, that's all laying the essential groundwork for those bigger organ replacements down the line.
Okay, so that's the direct therapeutic side. But our sources also stress this idea of dual utility using tissue engineering to create models in vitro. Why is building a tiny piece of human tissue on a chip So important.
Because it's a massive leap for translational medicine. Animal models are necessary, but let's be honest, they often don't fully replicate human disease or how we metabolize drugs.
Right, the number of drugs that work in mice but fail in humans is huge.
Exactly. So by creating accurate human disease models in vitro, whittle bits of functional tissue on a plate, we can study complex disorders like cancer progression or inflammatory conditions in a much more relevant setting.
And that speeds up drug development.
Hugely. These models become non-clinical testing platforms. You can screen potential drugs for efficacy and, just as importantly, for toxicity much faster and potentially more accurately. Imagine testing thousands of compounds on tiny human art muscle models to quickly weed out anything cardiotoxic.
Before it even gets near a patient.
Precisely. That saves enormous amounts of time and money in the drug development pipeline.
Okay, the potential seems enormous, almost limitless. But let's be realistic, many of these therapies aren't standard practice yet. What are the big roadblocks? The critical barriers slowing things down.
And we have to be pragmatic here. The field, while exciting, is still relatively young, and the barriers are significant and multi-layered. First, you've got major technical challenges.
Like what specifically?
Well, how do you reliably scale up production of these complex living tissues? How do you ensure consistency batch to batch? How do you guarantee that the engineered tissue integrates properly with the patient's own blood supply and nerves? These are not trivial problems.
Okay, technical hurdles. What else?
Second, and we'll definitely get into this more when we talk about stem cells, there are substantial safety issues. The biggest worry, frankly, is the risk of tumor formation, especially with certain types of stem cells.
Right, the tumorigenesis risk. We'll park that for later. And the final hurdle, often the trickiest one.
The regulatory burden. This is huge. These therapies, combinations of living cells, novel biomaterials, potent growth factors, they just don't fit neatly into the existing boxes for regulating drugs or medical devices.
So the rulebook hasn't caught up with the science.
Pretty much. Bringing a complex living product to market, especially something like an autologous therapy made individually for each patient, involves navigating completely new, often slow, and incredibly expensive regulatory pathways. That complexity and cost inevitably limits the speed of clinical impact.
Okay, let's dive into those core components you mentioned, starting with the cells. This is where it gets really fascinating, I think. And while stem cells grab all the headlines, you're saying the field actually started with more basic differentiated cells.
That's right. Somatic cells. These are just the normal, specialized working cells that we can harvest directly from a patient's existing tissues. They were the basis for the earliest, arguably simplest, success stories in the field.
Like the classic example of growing skin grafts for burn patients. Taking a small piece of their skin, expanding the cells in the lab, and making sheets to cover the burn.
Exactly that. Cultured epithelial autografts. Another key example is autologous chondrocyte implantation, or ACI, for cartilage defects, which we'll discuss more later. And the source material also highlights engineering functional bladder wall structures using a patient's own smooth muscle cells and uroepithelial cells.
And the massive advantage of using these somatic cells, initially at least, was
that they are autologous. They come from the patient themselves. That completely eliminates the risk of immunological rejection.
Which means no need for powerful immunosuppressant drugs.
Correct. And those drugs always come with their own package of side effects and long-term risks. So avoiding them is a major clinical plus.
But there must be a downside, otherwise we wouldn't have shifted so heavily towards stem cells. What's the limitation of using these already specialized cells?
It really comes down to numbers and flexibility. First, it can be hard to get enough of these specialized cells to start with. If you need to engineer a large piece of tissue, you need millions, potentially billions of cells.
And specialized cells don't like dividing indefinitely in the lab.
Exactly. They have a limited capacity to proliferate in vitro. They just won't expand to the massive numbers needed for many applications. Plus, they're already committed to being, say, a skin cell or a muscle cell. They lack the flexibility to become something else if needed. That limitation really drove the push towards finding a cell source that was both highly scalable and highly adaptable.
Which brings us squarely to the stem cell. So let's get the definition clear. What are the two absolute must-have characteristics of a stem cell?
Okay, fundamentally, they are undifferentiated, non-specialized cells. Their first critical ability is self-renewal. This means they can divide and make copies of themselves pretty much indefinitely when cultured under the right lab conditions.
So scalability.
Scalability, exactly. The second key feature is potency. This is their ability, when given the right signals, biochemical or physical cues to differentiate, to mature into one or more types of specialized cells that make up our body's tissues.
And the type of stem cell is often defined by how much potency it has, right? There's a hierarchy.
Yes, the potency spectrum is crucial. It dictates what the cell can potentially become and also carries different ethical considerations.
Let's quickly run through that. Top of the pyramid.
Totipotent cells.
Yeah.
These are the ultimate stem cells, like the fertilized egg, the zygote. They can form every cell type in the body, plus the extra embryonic tissues like the placenta.
Okay, one step down.
Pluripotent cells. These are typically derived from the inner cell mass of the early embryo, the blastocyst. They can differentiate into virtually any cell type of the body, proper muscle, nerve, bone, etc., but not the placenta. Embryonic stem cells fall here.
Got it. And the next level down.
Multipotent cells. These are often called adult stem cells or somatic stem cells, SSCs. They're found residing within various tissues in our bodies like bone marrow or the gut lining. Their job is normally to replace cells that die off or get damaged in that specific tissue. They are multipotent because they can differentiate, but only into a limited range of cell types, usually related to their tissue of origin.
Right. And within that multipotent category, the ones that have seen the most research and clinical use are the mesenchymal stem cells, or MSCs. But you mentioned earlier there was this nomenclature crisis. Why the name change from mesenchymal stem cell to mesenchymal stromal cell, even keeping the MSC abbreviation?
Yes. The ISCT, the International Society for Cellular Therapy, pushed for this change, and it was really important for scientific clarity and managing expectations. The original term, stem cell, strongly implied that their main therapeutic benefit came from direct differentiation.
Meaning the MSCs themselves turned into bone cells or cartilage cells to replace the damaged tissue.
That was the assumption. But as research progressed, a lot of evidence showed that in many situations, the major benefit wasn't coming from the MSCs differentiating themselves. Instead, it was coming from their paracrine and secretory activity.
Paracrine, meaning they release signaling molecules.
Exactly. They act like little local factories, pumping out growth factors, cytokines, anti-inflammatory molecules. These signals then influence the host's own cells and immune system, dampening inflammation, promoting blood vessel growth, and encouraging the native tissue repair mechanisms to kick in.
So they're more like conductors of the repair orchestra rather than playing all the instruments themselves.
That's a fantastic analogy, yes. The term stromal better reflects this supportive environmental influencing role, rather than just being a direct replacement part. It acknowledges they contribute indirectly. Getting this terminology right is vital for clear communication with clinicians, regulators, and patients about how these therapies are actually thought to work.
Okay, that makes sense. And to make sure researchers were all talking about the same population of cells, the ISCT set out some minimum criteria back in 2006 to define what qualifies as an MSC for research. What were those key criteria?
There were three main pillars. First, a basic physical characteristic. The cells have to stick to plastic when cultured in the lab. It sounds simple, but it's a typical feature of these stromal-type cells.
Okay, adherence to plastic.
Second, they need to display a specific pattern of surface markers, which is usually checked using flow cytometry. They must be positive for certain markers, typically CD105, CD73, and CD90. And just as importantly, they must be negative for a panel of markers associated with blood cells and immune cells.
Which negative markers specifically?
Things like CD45, a general white blood cell marker, CD34, hematopoietic stem progenitor cells, CD14 or CD11b, monocytes macrophages, CD79 or CD19 B cells, and HLA-DR, an immune activation marker.
That negative panel sounds like a crucial safety check.
It absolutely is. It's the assurance that you're not inadvertently injecting immune-reactive cells or blood precursor cells that could cause problems. That specific negative profile helps confirm they are likely immune-evasive or immune-modulatory, not immune-activating.
Okay. Plastic adherence, specific surface markers, positive and negative. What's the third pillar?
The third is functional. They must demonstrate trilineage differentiation potential in vitro. This means that in the lab dish, if you give them the right specific chemical cocktails, you have to show they can be coaxed into becoming osteoblasts, bone cells, adipocytes, fat cells, and chondroblasts, cartilage cells.
So even if they don't primarily work by differentiating in vivo, you still need to prove they retain that potential in the lab.
Correct. It's part of the defining characterization package.
And given that how these cells are sourced and handled can vary, there's also a standardized way to report on them using the DOSES framework. Can you quickly break down what DOSES stands for?
Sure. Doses is all about ensuring clarity and comparability in publications and clinical reports. It stands for
Key characteristics of the person the cells came from: age, health status.
which tissue were they isolated from? Bone marrow, adipose tissue, fat, umbilical cord, blood, etc.
How exactly were the cells isolated and purified?
What were their specific properties? Confirmation of the ISCT markers, viability percentages, maybe passage number, site of delivery. How and where were the cells administered to the patient? Intravenously, direct injection into a joint, seated onto a scaffold.
That level of detail seems critical if you want to compare results from different studies or manufacturers.
It's essential, because an MSC product derived from bone marrow might behave quite differently from one derived from fat, even from the same donor, depending on how it was processed and handled.
Right, and these MSCs, typically sourced from bone marrow or fat, offer flexibility in how they're used.
Yes, that's a key advantage. You can isolate them and potentially use them almost immediately, perhaps concentrated at the point of care during a surgery. Or, more commonly for larger scale applications, you can take that initial small sample and expand the cells significantly in vitro in a specialized lab, growing them up to large numbers. You could even start differentiating them towards a specific lineage using growth factors before they're delivered back to the patient.
Okay, so MSCs are the workhorse adult stem cells. Now let's shift gears to the truly pluripotent cells, the ones with maximum potential but also maximum baggage, starting with embryonic stem cells, ESCs.
Right. ESCs are derived from the inner cell mass of the very early human embryo, the blastocyst, usually around day 4 or 5 post-fertilization. Their strengths are undeniable. They have fantastic self-renewal capacity and that broad pluripotency, the ability to become almost any cell type.
But the baggage is significant. Two major limitations.
Exactly. First, the ethical concerns. Because deriving ESCs typically involves the destruction of human embryo, often surplus ones from IVF clinics, it has generated enormous ethical and political debate and restrictions in many parts of the world.
That's the ethical hurdle. What's the main clinical or biological one?
They are allogenic, meaning they come from a genetically different individual than the recipient. As a result, the recipient's immune system will recognize them as foreign and attack them, leading to graft rejection.
So just like an organ transplant you'd need.
Lifelong immunosuppression, powerful drugs to dampen the immune system, which, as we said, carry significant risks and side effects. This practical barrier greatly limits the widespread clinical use of ESCs, despite their potential.
Which leads us perfectly to the breakthrough technology that aimed to solve both of these problems. Induced pluripotent stem cells, iPSCs. This discovery by Yamanaka and Gurdon in 2006 really turned the field on its head.
It was absolutely revolutionary, game-changing. What they showed was that you could take a mature, specialized adult cell, something as simple as a skin fibroblast or even a blood cell, and essentially wind back its developmental clock. You could reprogram it back to an embryonic-like pluripotent state.
Making it behave like an ESC, but without using an embryo.
Precisely. The key insight was that introducing just a handful of specific regulatory genes, known as transcription factors, could trigger this complete cellular identity reset.
And the famous Yamanaka factors or OSKM factors are
OCT34, SOX2, KLF4, couple-like factor 4, and CMYC. Initially, they delivered the genes encoding these factors into the adult cells using viral vectors.
like retroviruses or lentiviruses.
Yes. And those viruses are very efficient at getting the genes in, but they carry a risk because they integrate into the host cell's genome.
And if they integrate in the wrong place.
If they happen to insert near a gene that controls cell growth, an oncogene, they could potentially activate it, leading to uncontrolled proliferation, basically, causing cancer. That risk of insertional mutagenesis was a major safety concern with early viral methods.
So the field has moved towards safer ways to deliver these reprogramming factors.
Definitely. There's been intense research into non-integrating methods. This includes using viruses that don't insert into the genome, or avoiding viruses altogether by delivering the factors transiently using synthetic messenger RNA, mRNA, or even cocktails of specific small molecule drugs that can mimic the effect of the transcription factors.
These non-viral, non-integrated methods are becoming the standard for clinical-grade iPSC production then.
They are, yes, because they dramatically reduce that theoretical cancer risk associated with genomic integration.
Okay, so iPSCs potentially solve the ethical issue of ESCs. What about the immune rejection problem?
This is the other huge advantage. Because you can generate iPSCs directly from the patient who needs the therapy taking their skin or blood cells. The resulting pluripotent cells are genetically identical to that patient. They are autologous.
Ah, so when you differentiate those iPSCs into the target cell type, say heart muscle cells, and transplant them back into the same patient.
There should be no immune rejection, no need for immunosuppression. It potentially combines the best of both worlds, the pluripotency of ESCs with the immune compatibility of autologous somatic cells.
That is genuinely a massive leap. It overcomes the two biggest hurdles of ESCs.
It really does. They proliferate efficiently like ESCs. They have that broad differentiation potential. They sidestep the main ethical concerns, and they offer that autologous rejection-free possibility.
You also mentioned banking, even if autologous isn't always feasible.
Right. For situations where making patient-specific iPSCs might be too slow or costly, you can still create banks of iPSCs from carefully screened volunteer donors. These banks can be characterized by their HLA type, the immune system markers. So you could potentially find a close enough HLA match for many patients, reducing, though maybe not always eliminating, the need for heavy immunosuppression. It offers another route to broader application.
Okay, so we have these incredibly powerful pluripotent cells, either ESCs or, more likely now, iPSCs, but they're still just blank slates. The final crucial step before they can be used therapeutically is getting them to turn into the specific cell type you need, right? The in vitro differentiation.
Exactly. And this is where developmental biology meets cell culture engineering. It's incredibly complex and requires very precise control over the cell's environment.
What does that involve?
It means moving away from simple culture media with serum to highly defined, bespoke, chemically defined media. These contain precise cocktails of small molecules, cytokines, and specific growth factors that are added or removed at very specific times during the culture process.
So you're basically trying to mimic the sequence of signals that happens during natural embryonic development.
That's the goal. You're trying to guide the cells down a specific differentiation pathway, say, towards becoming chondrocytes for cartilage repair, or cardiomyocytes for heart muscle, or neurons for neurological applications by activating and inhibiting the right signaling pathways in the right order.
It sounds incredibly sensitive.
It is, and it's not just about the chemical soup they're swimming in. You mentioned earlier that the physical environment matters just as much.
Ah, yes, the materials and mechanics.
Absolutely. Cell fate isn't just directed by soluble factors. The physical properties of the surface the cells are growing on, the stiffness or elasticity of the scaffold material they might be embedded in, even the application of mechanical forces like stretch or fluid flow. All these physical cues play a critical role in telling a stem cell what to become.
So successful differentiation requires managing both the chemical recipe and the physical architecture simultaneously.
Precisely. You need to get both the biochemical signaling and the biophysical cues right to reliably generate functional, mature cells of the desired type at scale. It's a major ongoing challenge in the field.
Okay, let's ground this in a real-world clinical example. You mentioned articular cartilage earlier, that smooth tissue lining our joints that wears out in arthritis and is notoriously bad at healing itself. Let's compare the traditional surgical approach with the regenerative one.
Right, cartilage is a perfect example because it really highlights that difference between basic repair and true regeneration. The traditional mainstay for smaller cartilage defects for, over 30 years now, has been a technique called microfracture.
Microfracture, what does that involve? Sounds like drilling.
Pretty much. The surgeon uses a sharp tool to create small holes or fractures in the bone just underneath the cartilage defect, the subchondral bone. The idea is to create channels that allow bleeding into the defect site.
And the blood brings
cells from the bone marrow, specifically marrow stromal cells, which contain those mesenchymal stem stromal cells we discussed, along with blood clotting factors. These form a clot in the defect, which then sort of organizes into a repair tissue.
So the body is triggered to form its own patch, essentially. Sounds simple enough. What's the catch?
The catch is the type of tissue that forms. It's not the smooth, resilient, highly organized hyaline cartilage that normally lines the joint. Instead, you get fibrocartilage.
Fibrocartilage, and that's not as good.
Not nearly as good, especially long-term. Fibrocartilage is structurally different, more like scar tissue. It's mechanically inferior, less organized, and lacks the durability of native hyaline cartilage. It might provide some initial cushioning and pain relief, but it tends to break down over time, leading to the symptoms returning. It's a repair, but often not a lasting one.
Okay, so microfracture gives you fibrocartilage, which isn't ideal. This limitation is what drove the development of autologous chondrocyte implantation, ACI, right? Which aims for true hyaline cartilage.
Exactly. ACI is a much more involved two-stage regenerative approach, specifically designed to promote the formation of hyaline-like cartilage.
2 stages. What's the first stage?
Stage 1 involves arthroscopy keyhole surgery. The surgeon inspects the joint, confirms the defect is suitable for ACI, and takes a very small biopsy of healthy cartilage from a non-weight-bearing area of the same knee. We're talking about a piece maybe the size of a Tic Tac or a grain of rice.
Tiny. And what happens to that biopsy?
It gets sent off to a highly specialized laboratory that operates under good manufacturing practice, GMP conditions, very strict quality control. In the lab, the cartilage cells, the chondrocytes, are enzymatically released from the matrix and then cultured and expanded over several weeks.
Expanding them, meaning growing many more copies.
Yes, dramatically increasing the number of healthy chondrocytes.
Yeah.
You go from maybe a few 100,000 cells in the biopsy to many millions.
Okay, so that's stage 1, harvest and expansion. What's stage 2?
Stage 2 is the reimplantation, and this usually requires a second, often open, surgical procedure. The surgeon prepares the cartilage defect again and then implants those millions of cultured, healthy autologous chondrocytes back into the cleaned-out defect.
How do they keep the cells in place? They're just loose cells at this point, aren't they?
Good question. Typically, the cells are injected under a patch or membrane that the surgeon stitches or glues over the defect. This could be a piece of periosteum, the membrane-covering bone, taken from the patient, or increasingly, a commercially available collagen membrane. This patch acts like a little biological container, holding the cells in the defect while they start producing their own extracellular matrix and forming the new cartilage tissue over months.
Wow. That sounds significantly more complex, time-consuming, and presumably expensive than just drilling a few holes like in microfracture.
Oh, absolutely. 2 surgeries, specialized lab work, GMP conditions. It's a much bigger undertaking.
And yet, you mentioned the UK's National Institute for Health and Care Excellence, NIST-CE, which is notoriously tough on cost-effectiveness, actually endorsed ACI for certain types of knee cartilage defects back in 2017. Why did this complex, expensive regenerative approach get the green light?
It came down to the evidence showing a superior and more durable clinical outcome. Studies comparing ACI to microfracture found that ACI resulted in repair tissue that was structurally closer to hyaline cartilage and provided better, longer-lasting functional improvement for patients with specific types of larger defects.
So the quality of the repair justified the cost?
Essentially, yes. NEET looked at the cost per quality-adjusted life year, QALY gained. This is a standard metric used in health economics that considers both the length and the quality of life improvement. They determined that for suitable patients, the cost per QALY gained with ACI was less than their typical threshold, which was around 20,000 to 30,000 pounds at the time.
So it was deemed cost-effective because it provided a significantly better, more lasting functional result compared to the simpler but less effective microfracture approach.
Exactly. ACI is a great example, proving that if a regenerative therapy can genuinely deliver a superior, durable biological outcome, the initial higher complexity and cost can be justified from both a clinical and an economic perspective. It sets a precedent.
Okay, so we've talked about the cells, the seeds of regeneration. Now let's talk about the materials, the soil, the scaffold, the environment they live in. You said these aren't just passive supports.
Not at all. The materials or scaffolds are critically important. They do provide structural support, of course, shaping the new tissue as it forms, but they also act as delivery vehicles for cells or molecules, and increasingly, we understand they provide crucial physical and chemical cues that actively influence cell behavior, things like adhesion, migration, proliferation, and differentiation. They're active participants.
Right. Let's start with a category surgeons are very familiar with, synthetic and engineered polymers. We use these all the time.
We certainly do. We use non-resorbable polymers for permanent implants, things like PMMA, polymethyl methacrylate, which is the acrylic bone cement used in joint replacements, or materials like Dacron or PTFE used for synthetic vascular grafts that need to last a lifetime.
But for tissue engineering, where you want the body to eventually replace the scaffold, you're mostly interested in resorbable polymers.
Precisely. These are polymers designed to degrade and be absorbed by the body over time, ideally at a rate that matches the formation of new tissue by the infiltrating or delivered cells. We're talking about materials like PCl, polycaprolactone, PLA, polylactic acid, PGA, polyglycolic acid, and their copolymers like PLGA, polylactic co-glycolic acid.
The same materials used in dissolvable stitches.
Exactly the same family. Their history in sutures and orthopedic devices means their degradation characteristics and biocompatibility are really well understood. That makes them attractive starting points for creating scaffolds.
But you can't just implant a solid block of PLA, can you? Cells need space to grow, nutrients need to get in. How do you turn these basic polymers into functional scaffolds?
You need to create porosity, an interconnected network of pores that allows cells to penetrate, attach, and receive nutrients, while also allowing waste products to diffuse out. There are various manufacturing techniques to achieve this.
Like what?
Well, one approach is to create porous monoliths, sort of like sponges, using methods like gas foaming, salt leaching, or phase separation. Sometimes high-pressure gases like supercritical carbon dioxide are used to create very fine, controlled pore structures.
Okay, so making sponge-like structures. What else?
A more advanced and increasingly popular technique for creating fibrous scaffolds that mimic the natural extracellular matrix is electrospinning.
Electrospinning. That sounds high-tech. How does that work? It create fibers, you said.
Yeah, it's a fascinating process. You dissolve the polymer in a solvent to make a solution, which is then loaded into a syringe and pumped through a fine needle. Critically, a very high voltage, we're talking kilovolts, is applied between the needle tip and a collector plate some distance away.
Okay, high voltage, what does that do?
The strong electric field puts a charge on the surface of the polymer solution droplet at the needle tip. When the electrostatic repulsion overcomes the surface tension, a jet of the polymer solution erupts and accelerates towards the grounded collector plate. As the jet travels through the air, the solvent rapidly evaporates,
Leaving behind
a very thin, continuous fiber of solid polymer. Because the jet path is chaotic due to bending instabilities, you end up collecting a non-woven mat, or mesh, composed of these incredibly fine fibers on the collector plate.
How fine are we talking?
The fiber diameters can range from micrometers down to nanometers, which is really important.
Why is that scale so crucial for tissue engineering?
Because the natural extracellular matrix, ECM, that surrounds cells in our tissues is also composed of nanoscale fibers, like collagen and elastin. Electrospun scaffolds, with their incredibly high surface area to volume ratio and fiber structure mimicking the native ECM, provide excellent physical cues for cell attachment, alignment, and growth.
So the cells feel more at home on these structures.
That's the idea. They provide topographical guidance. Electrospinning is being heavily explored for creating scaffolds for various tissues, including skin, nerve conduits, blood vessels, and, as mentioned in the source, for things like reinforcing rotator cuff repairs in the shoulder.
Okay, so that covers polymers. Let's move to a different class, bioceramics. Specifically, the example given is bioglass. What's special about this material? It's not crystalline, you said.
Right. Bioglass is an amorphous material, a glass typically composed of oxides of silicon, sodium, calcium, and phosphorus. N2O, Ke2O5, SiO2. What makes it really special is its bioactivity. When it's placed in contact with body fluids, it doesn't just sit there passively.
It interacts chemically.
Yes, it undergoes a series of surface chemical reactions. It starts to slowly dissolve, leaching out its component ions, sodium, calcium, phosphate, and silicate ions into the surrounding environment.
And what effect does that have?
It triggers two really important responses. First, the release of calcium and phosphate ions, combined with changes in local pH, leads to the precipitation of a calcium phosphate layer on the bioglass surface. This layer is very similar to the mineral component of bone, hydroxyapatite.
So it forms a bone-like layer.
Exactly. And this hydroxycarbonate apatite, HCA layer, allows the bioglass to form a strong chemical bond directly with the host bone tissue, and interestingly, also with soft tissues. It doesn't just rely on mechanical interlocking. It chemically integrates.
That's the bonding aspect. What was the second crucial response?
The second part is the biological signaling. The ions that are released, particularly the silicate and calcium ions, have been shown to have specific biological effects on cells. They act like signaling molecules.
How so?
They can stimulate key intracellular signal pathways in surrounding cells, like osteoprogenitor cells and mesenchymal stem cells. This can upregulate the expression of genes involved in bone formation, essentially recruiting the body's own regenerative cells to the site and encouraging them to differentiate and lay down new bone.
So bioglass isn't just a scaffold, it's actively stimulating the body's repair process.
That's right. It's osteoconductive, provides a scaffold, and osteoinductive stimulates new bone formation. This unique combination of bonding and bioactivity makes it very useful in bone grafting applications, often used as granules, putties, or even porous foamed structures.
And the source mentioned toothpaste.
Yeah, a derivative form of bioglass, often called novavamin, is used as an additive in some remineralizing toothpastes. The idea is that it deposits that calcium phosphate layer onto exposed dentine to block tubules and reduce sensitivity, and potentially helps remineralize early enamel lesions.
Fascinating. Okay, from hard ceramics to something much softer, hydrogels. These seem intuitively suited for working with living cells.
They really are. Hydrogels are essentially networks of hydrophilic, water-loving polymer chains dispersed in a large amount of water. They can be mostly water, sometimes up to 99% by weight, yet they maintain a solid gel-like structure, kind of like Jell-O or contact lenses.
And why is that high water content so beneficial for tissue engineering?
Because it creates an environment that's very similar to the natural extracellular matrix of many soft tissues. More importantly, that high water content allows for very efficient diffusion. Nutrients, oxygen, and growth factors can easily diffuse into the gel to reach encapsulated cells, and waste products from the cells can easily diffuse out.
So cells trapped inside stay happy and viable?
Generally, yes. Hydrogels provide a protective, hydrated environment that supports high cell viability, which is why they're widely used for cell encapsulation and delivery.
And the example given was using alginate hydrogel to encapsulate pancreatic islets.
Right. Alginate is a natural polysaccharide derived from seaweed. It can be easily cross-linked, often using calcium ions, to form a stable hydrogel around cells like pancreatic islets. The idea here is often immunoisolation.
Meaning the gel protects the islets from the recipient's immune system.
Exactly. If you're transplanting islets from a donor, allogeneic islets, to treat diabetes, the alginate capsule can act as a physical barrier, preventing immune cells and large antibodies from reaching and destroying the islets. However, the gel remains porous enough for small molecules like glucose to diffuse in, and crucially, for the insulin produced by the islets to diffuse out into the bloodstream.
Clever. But you hinted earlier that traditional hydrogels have a drawback, something about mechanical properties.
Yes, a major challenge with many simple hydrogels is that they tend to be mechanically weak, especially during the gelation process or before they are fully cross-linked. They can be difficult to handle, they might collapse under their own weight, and it's hard to create precise, complex 3D structures with them using traditional methods.
So how are researchers overcoming this fragility to build more complex hydrogel-based tissues? This leads to advanced manufacturing, like 3D bioprinting.
Exactly. Standard 3D printing struggles with printing very soft materials like low-viscosity hydrogel precursors because they just slump and spread before they can solidify. So several innovative techniques have been developed to provide temporary support during the printing process.
Like the fresh-ish technique.
Right. FRESH stands for Freeform Reversible Embedding of Suspended Hydrogels. The core idea is to print the bioink, the hydrogel precursor containing cells, into a support bath. This bath isn't liquid. It's made of a tightly packed gelatin microparticle slurry.
Gelatin, like Jell-O again.
Kind of. This gelatin slurry behaves like a Bingham plastic. It acts like a soft solid at rest, holding the printed structure in place, but it yields and flows like a liquid locally where the printing needle moves through it. So you can print complex structures within this supportive bath.
And how do you get the printed structure out afterwards?
That's the reversible embedding part. Gelatin melts at body temperature, 37 degrees C. So once the printed hydrogel construct has solidified, e.g. by UV cross-linking or chemical reaction, you simply warm the whole thing up gently. The gelatin support bath melts away into a liquid, leaving your freestanding, complex hydrogel structure intact.
Very neat. And the other technique mentioned was SLAM.
Yes, SLAM, or suspended layer additive manufacturing. It's conceptually similar in using a support bath, but the bath here is typically composed of hydrogel microparticulates that are shear thinning. Again, the bath holds the printed structure, allowing high-resolution printing.
And removal, is it also temperature-based?
With SLAM, the removal is often even simpler. The support bath particulates are typically designed so they can be easily washed away with water or a simple buffer solution once the main hydrogel construct is stable. No heating required.
What kind of structures can be made with these advanced techniques?
They allow for really high-resolution printing of intricate shapes that would be impossible otherwise. The sources mention examples like printing branching structures, mimicking carotid arteries, or even creating multi-material constructs like an osteochondral plug for cartilage repair that has a distinct bony layer made of one hydrogel composite and a cartilaginous layer made of another, all printed within the same structure. It opens up possibilities for much more biomimetic and complex tissue designs. Okay, so we've got the cells, the workers, and the materials, the scaffold. The third piece of the puzzle is the molecules. These are the chemical signals, the instructions, used to fine-tune the whole process. Exactly. The goal here is to use specific molecules, often growth factors, cytokines, small molecule drugs, or even things like therapeutic RNAs, to deliberately modify the tissue environment. We want to either enhance the regenerative processes or, just as importantly, suppress negative outcomes like excessive inflammation, fibrosis, or scar formation.
Right. Let's look at the first case study you mentioned. Trying to prevent excessive skin scarring after injury, a major issue in surgery and burns. What's the normal process that goes wrong in bad scarring?
Okay, so normally after skin injury, platelets arrive, release clotting factors, but also a whole host of pro-inflammatory signals, TNF, various interleukins, ILs, and growth factors like PDGF and VEGF. This calls in inflammatory cells and fibroblasts.
And fibroblasts start making?
Extracellular matrix. Initially, they lay down collagen type 3, which is relatively weak. A key signaling molecule here is TGF-1, transforming growth factor beta-1, which strongly promotes matrix production. Over time, this initial matrix gets remodeled into stronger, more organized collagen type 1, leading to a functional minimal scar.
So where does it go wrong in severe scarring or hypertrophic scars?
The pathology arises when the initial damage is very severe or that prolonged inflammation may be due to infection. This leads to an inflammatory overshoot. You get excessive and prolonged release of pro-inflammatory signals and particularly very high levels of TGF-1.
And too much TGF-1 leads to?
An over-recruitment and over-activation of specialized fibroblasts called myofibroblasts. These cells are highly contractile and produce excessive amounts of disorganized collagen. The result is that thick, raised, often tight and inelastic scar tissue, hypertrophic scars or keloids that we want to avoid.
Okay, so the problem is excessive TGF-1 signaling. What was the molecular intervention strategy?
The strategy focused on using a different member of the TGF family, TGF-3.
How does TGF-3 help?
TGF-3 acts as a natural regulator or antagonist in this system. It can bind to the same cell surface receptors as TGF-1 and its close relative TGF-2, but it triggers a different downstream signal. Essentially, it competes with and blocks the profibrotic signaling of TGF-12. By applying TGF-3 locally to the wound, the idea was to modulate that excessive signaling, regulate the matrix formation, and encourage a more normal, less fibrotic healing process.
And did it work? The source says it showed promise in early clinical trials.
Yes, phase 1 and 2 trials suggested it was effective in reducing scar formation compared to placebo. The science seems solid.
But it didn't become a standard treatment. Why not? This seems like a crucial lesson.
It's a classic and harsh lesson in translational challenges, particularly around manufacturing and cost. TGF-3 is a protein, a relatively large biomolecule. Manufacturing proteins to the extremely high purity and consistency required for clinical use, GMP grade, is technically complex and very, very expensive compared to producing traditional small molecule drugs.
So the cost of goods was just too high.
Exactly. The projected cost of the final TGF-3 treatment was simply too prohibitive for widespread clinical adoption, especially for something like scar prevention, which, while important, might be seen as less critical than life-saving therapies. Despite the promising science, the economics just didn't work out. It highlights that technical feasibility isn't enough. You also need economic viability for clinical translation.
A sobering example. Okay, let's look at the second case study. Preventing ocular fibrosis, specifically scarring in the cornea after infection, which can lead to blindness. The therapeutic agent here was different, a proteoglycan called Paracrine.
Right. Corneal transparency is essential for vision, and it depends on the highly organized parallel arrangement of collagen fibers in the corneal stroma. After infection or injury, the repair process often involves rapid but disordered collagen deposition, leading to clouding and vision loss.
And decorin helps prevent this disordered deposition. How?
Decorin is a fascinating molecule because it plays a structural role and a signaling role. Structurally, in healthy cornea, decorin molecules naturally bind to the surface of collagen type I fibrils, helping to regulate their diameter and spacing, contributing to that crucial ordered architecture.
Okay, so it helps organize collagen normally. What about its signaling role in fibrosis?
When fibrosis is kicking off, again driven largely by TGD1, free decorin molecules, not bound to collagen, act like molecular sponges. Decorin has a known affinity for binding to several key growth factors and cytokines, including, crucially, TGF-1.
So it mops up the excess TGF-1.
Precisely. By binding to and sequestering TGF-1, decorin effectively neutralizes the main profibrotic signal, driving that disordered collagen production by corneal fibroblasts, keratocytes.
A very elegant mechanism. And how is it delivered in the studies?
The solution was ingeniously simple and practical. They incorporated Paracrine into an eye drop formulation. This allowed for localized, sustained delivery directly to the corneal surface.
And the results?
In preclinical models of corneal injury, the Paracrine eye drops were shown to be effective. They significantly reduced the expression of key scar markers, like alpha-smooth muscle actin, SMA, which indicates myofibroblast formation, and fibronectin, another matrix protein associated with scarring. Importantly, the treatment helped restore the normal, transparent structure of the cornea.
That sounds much more promising from a translational perspective than the TGF-3 story and eye drop is a familiar, relatively inexpensive delivery method.
Absolutely. It illustrates how choosing the right molecule and finding a practical, cost-effective delivery strategy are both critical for success.
But both examples really hammer home the challenge of molecular delivery in general, don't they? Getting the molecule to the right place at the right concentration for the right amount of time?
It's a central challenge in the field. Sustained, localized release is almost always required. Small molecule drugs tend to diffuse away very quickly from the site of administration, so you need clever encapsulation strategies within materials or hydrogels to achieve slow, controlled release over days or weeks.
And it's even harder for large biomolecules like growth factors or proteins.
Yes, significantly harder. Large biomolecules like TGF-3 or Paracrine are often very sensitive to their environment. Changes in pH, temperature, or exposure to enzymes in the body can rapidly degrade them or cause them to lose their biological activity. So the delivery system, the scaffold or hydrogel, not only has to control their release rate, but also protect their structure and function. Plus, as we saw with TGF-3, their high intrinsic production cost means you absolutely have to maximize their potency and minimize waste, which puts enormous demands on the sophistication of the delivery vehicle. Getting molecular delivery right is arguably one of the biggest hurdles for many regenerative therapies.
Right, we absolutely have to address the safety aspects. As these powerful therapies move closer to routine clinical use, being vigilant about the potential risks is paramount.
And looking at the potential safety concerns, there's one that really stands out as the most serious, particularly linked to those highly potent, pluripotent cells, the ESCs and iPSCs. Without a doubt, that's tumorigenesis, the risk of the transplanted cells forming tumors. Specifically, the major concern with pluripotent stem cells is the formation of teratomas.
Teratomas, what are they exactly?
A teratoma is a specific type of tumor that's actually composed of a disorganized mixture of tissues derived from all three embryonic germ layers: ectoderm, mesoderm, endoderm. You might find bits of hair, teeth, muscle, neural tissue all jumbled together. The ability to form teratomas when injected into immunodeficient animals is, ironically, one of the defining tests used to confirm pluripotency in the lab.
So the very power that makes them useful also makes them dangerous if not properly controlled.
Exactly. The danger lies in inadvertently transplanting even a small number of residual undifferentiated pluripotent stem cells, along with the intended differentiated cells. These residual pluripotent cells could then potentially survive and proliferate uncontrollably in the patient, forming a teratoma.
That sounds like a major barrier. What are the main strategies being used to mitigate this fundamental risk?
Mitigation is multi-pronged. The first and most critical line of defense is rigorous quality control of the final cell product before transplantation. This involves developing highly efficient and robust in vitro differentiation protocols that drive virtually all the cells to the desired mature state.
So proving you've gotten rid of all the undifferentiated ones?
Yes, using sensitive assays to confirm the purity of the final differentiated cell population, and crucially, to demonstrate the complete absence, or at least reduction below a defined safety threshold, of any remaining undifferentiated pluripotent stem cells, often identified by specific markers like TRA160 or SEA4.
Okay, so better differentiation and purification. What else?
The second strategy relates back to how the iPSCs were made in the first place. As we discussed, moving away from integrating viral vectors towards non-integrating viral methods or entirely non-viral reprogramming techniques like mRNA or small molecules significantly reduces the separate but related risk of insertional mutagenesis causing cancer.
Right, avoiding messing with the genome. Is there a third approach that avoids pluripotency altogether?
Yes, and that's the concept of transdifferentiation, also known as direct reprogramming. This is a really exciting area, though still largely experimental. The idea is to develop methods to convert one type of mature, specialized somatic cell directly into another type of specialized somatic cell without going through the intermediate pluripotent state.
Like turning a skin cell directly into a functioning neuron or a heart muscle cell.
Exactly. If you could achieve that efficiently and reliably, you would completely bypass the pluripotent stage and therefore eliminate the associated teratoma risk. It's technically very challenging, but it represents perhaps the ultimate safety goal for cellular reprogramming.
Okay, so tumorigenesis is the big one for pluripotent cells. What about therapies using allogeneic cells from an unrelated donor, even if they're just MSCs or differentiated cells, not pluripotent ones? What are the risks there?
Two main risks with allogeneic cells. First, the potential transmission of infection. Anytime you're transferring cells or tissues from one individual to another, there's a risk, however small, of transmitting known or even unknown infectious agents. Viruses, bacteria, prions.
How is that risk managed?
Through extremely rigorous donor screening protocols, testing donors for a wide range of pathogens, and critically, by ensuring that all cell processing, expansion, and tissue engineering steps are performed under the highly controlled, sterile conditions mandated by Good Manufacturing Practice, GMP regulations. GMP is essential for minimizing contamination risk.
Okay, infection control via GMP. What's the second allogeneic risk?
Immune rejection. Even if the cells aren't pluripotent, if they come from a genetically different donor, the recipient's immune system is likely to recognize them as foreign and mount an attack, destroying the graft.
Even with things like MSCs, which are sometimes described as immune privileged.
MSCs do have immunomodulatory properties, and they express low levels of MHC class I and lack MHC class II, which helps them evade immediate rejection. However, long-term survival and function of allogeneic MSCs without any immunosuppression is still debated and likely context-dependent. For other allogeneic differentiated cell types, rejection is a near certainty without intervention.
So just like with solid organ transplants, allogeneic cell therapies often still require
Some degree of immunosuppression for the patient. While HLA matching, trying to match the immune markers between donor and recipient, can help reduce the intensity of rejection, it rarely eliminates it completely. So the patient may need to take immunosuppressive drugs long-term, with all the associated risks of infection and other side effects. This burden of immunosuppression remains a significant hurdle for many allogeneic approaches. Understood. Okay, let's pivot finally to the future. Where is this field heading in the next, say, 5 to 10 years? What are the exciting trajectories? Well, the pace of innovation is certainly not slowing down. We're seeing the emergence of several potentially transformative technologies. One is harnessing extracellular vesicles, EVs, for therapeutic signaling.
EVs like exosomes, little packages cells release.
Exactly. Cells naturally release these tiny membrane-bound vesicles that are packed with proteins, lipids, and nucleic acids, like microRNAs. They act as messengers, carrying signals between cells. The idea is that we might be able to isolate EVs from therapeutic cells, like MSCs, and use the EVs themselves as a cell-free therapy, or even engineer EVs to carry specific therapeutic cargo.
So getting the therapeutic effect without transplanting the whole cell.
Potentially, yes. It could offer safety advantages. Another huge area is, of course, gene editing, particularly using tools like CRISPR-Cas9.
How does CRISPR fit into tissue engineering?
In multiple ways. You could use gene editing to correct genetic defects in patient-derived iPSCs before differentiating them for treating inherited diseases. Or you could edit the cells to make them safer, perhaps adding suicide genes that could be activated if they started forming tumors. Or even edit them to make them less immunogenic, potentially creating universal donor cells that wouldn't be rejected. The possibilities are vast, though obviously ethical and safety considerations are paramount.
And all this increasing complexity points towards more personalized approaches.
Absolutely. The future is undoubtedly heading towards precision medicine in this space. We'll move away from one-size-fits-all therapies. Instead, treatment will likely involve detailed patient stratification based on their specific molecular profile, their genetics, the specific phenotype of their disease, maybe biomarkers indicating their likely response.
Tailoring the regenerative strategy to the individual patient.
It's exactly which regenerative cells to use, which scaffold material, which molecular cues the optimal combination might differ significantly between patient subgroups. This level of personalization demands much more sophisticated diagnostics and also requires our regulatory frameworks to evolve rapidly.
How so?
Regulators need to develop clear pathways for approving these highly complex, often personalized, advanced therapy medicinal products, ATMPs. This includes figuring out how to regulate advanced manufacturing platforms, especially things like automated, closed-system point-of-care manufacturing, where a patient's cells might be processed and the therapy prepared right there in the hospital. Ensuring safety, efficacy, and consistency in these decentralized models is a major challenge for regulators worldwide. #TagOutro.
Wow. Okay, this deep dive has really taken us across the entire landscape of tissue engineering. It seems clear that this journey, from just repairing tissues to actually regenerating them, truly hinges on mastering that intricate, controlled interplay between the cells, the materials, and the molecules.
It really does. And the key to controlling that interplay, the secret sauce if you like, is understanding and harnessing the fundamental principles of developmental biology. We're essentially trying to recapitulate in a controlled way in the lab or in the patient the same complex signaling events and environmental cues that nature uses to build tissues perfectly during embryonic development.
And this knowledge isn't just for lab researchers, is it? It feels incredibly relevant for surgeons on the front line.
Absolutely. Surgeons are increasingly at the center of this field. They're involved in harvesting the patient's cells, implanting the engineered constructs, assessing the outcomes, and often collaborating directly in the translational research developing these therapies. Understanding the science behind these approaches is becoming essential for modern surgical practice.
Definitely. Now, thinking back to that example of TGF-3 for scar prevention, a scientifically promising molecule that failed translationally, mainly due to its huge manufacturing cost and complexity, it raises a difficult question. When we have these potentially life-changing or at least function-restoring regenerative treatments that face enormous hurdles just getting to patients because of cost and regulatory complexity, what role should government or regulatory bodies play? Should there be mechanisms to prioritize clinical potential and accessibility over purely commercial viability, especially for conditions with high unmet need?
That's a really tough one.
How do we structure the system, the funding, the regulation, the reimbursement to ensure that these incredible biological advances actually reach the patients who need them and aren't just left on the shelf because the initial economics are too challenging? That balance between innovation, safety, cost, and access. That's the provocative thought we'll leave you with today.